Discuss the bioinformatics. Now, first we discuss the introduction. It is an interdisciplinary research area of the biology and the computational biology. First, we discuss what is bioinformatics and its definition. Bioinformatics is a branch of science that integrates the computer science, mathematics, statistics, chemistry, engineering for analysis, exploration, integration, and exploitation of the biological sciences data in the research and the development. Bioinformatics deals with the storage, retrievals, analysis, and interpretation of the biological data using computer-based softwares and the tools. So, how bioinformatics is differ from a term computational biology. Basically, computational biology and bioinformatics use alternatively, but there is some difference, a basic difference between this. Bioinformatics is limited to the sequence, structure, and the functional analysis of genes and genomes and their corresponding product, whereas computational biology encompasses all the biological area that involve the computation. Example, mathematical modeling of ecosystem, population dynamics, application of game theory in behavioral study. बायोइन्फॉर्मेटिक्स का ये एक फ्लोचार्ट टाइप में है जिसमें कि वो किस किस ब्रांच से किस किस सेक्टर से वो पूरा कंबाइंड है वो दिखाया गया है. बायोइन्फॉर्मेटिक्स में जेनेटिक इन्फॉर्मेशन को पहले मॉलिक्यूलर स्ट्रक्चर में कन्वर्ट कर जेनेटिक इन्फॉर्मेशन कंप्यूटर्स के अंदर स्टोर होती है. वो मॉलिक्यूलर स्ट्रक्चर में कन्वर्ट होकर उसमें बायोकेमिकल फंक्शंस उसका देखा जाता है और उससे होने वाली डिजीज़ में जो फिनोटिपिक है जो सिम्टम्स हम देख सकते हैं वो हम इसमें डिटरमाइन कर सकते हैं बाय द यूज ऑफ द बायोइन्फॉर्मेटिक्स. Come to the history of bioinformatics, the term given by the Pauline Harvey and Van Hasper in 1970, first introduced in 1990. It dealt with the management and the analysis of the data pertaining to DNA, RNA, and protein sequence. 1972, Paul Wurzburg made the first recombinant DNA molecule using the ligase. That same year, Steinel Cohen produced the first recombinant DNA organism. Joseph Sambrock led a team that refined the DNA electrophoresis using the agarose gel in 1975. In 1977, a method for sequencing DNA was discovered. 1978, restriction fragment length polymorphism is discovered. 1980, PCR was invented by the Carl Mollius. In 1982, Genetics Computer Group was created. In 1986, Swiss Spot database was created in the Department of Medical Biochemistry of the University of Jena and the European Molecular Biological Laboratory. 1987, the use of yeast artificial chromosome was described. In 1987, Human Genome Initiative was started. In 1990, the BLAST program was implanted. 1991, first time the term bioinformatics appeared in the scientific literature. 1992, genetic linkage map of the entire human genome was published. In 1996, the cloned sheep Dolly was born. 1998, the Swiss Institute of Biotechnology was established a non-profit foundation. 1999, human chromosome was completely sequenced. In 2000, international research was sequenced. In 2001, human genome was published. In 2003, human genome project was completed in April 2003. In 2005, the genome sequence of rice was completed. Here is a evolution on database technology. In 1960s, data collection, database creation, IMS and network DMS. In 1970, relational data models was there. In 1980s, RDMBS, that is the Retrieval Database Management System there. In 1990s, data mining, data warehousing, web databases are there. In 2000, stream data management and mining was there. Data explosion, our genetic identity is encoded in long molecule which are made up of only the four basic units. These are the adenine, cytosine, guanine and thymine. Bioinformatic approaches, there are the three major areas: sequence analysis, structure analysis and the functional analysis. And the three divisions of bioinformatics are molecular bioinformatics, cellular and subcellular bioinformatics, organism and the community bioinformatics. Now application of bioinformatics are the structure analysis, में हम न्यूक्लिक एसिड स्ट्रक्चर का प्रिडिक्शन कर सकते हैं, प्रोटीन स्ट्रक्चर का प्रिडिक्शन हो सकता है, प्रोटीन स्ट्रक्चर क्लासिफिकेशन हो सकता है, प्रोटीन स्ट्रक्चर कंपैरिजन हो सकता है। सीक्वेंस एनालिसिस में जीनोम कंपैरिजन, फाइलोजेनी हम देख सकते हैं, उसमें फाइलोजेनेटिक ट्री का कंस्ट्रक्शन हो सकता है, जीन एंड प्रमोटर प्रिडिक्शन हो सकता है, मोटिव डिस्कवरी हो सकती है, सीक्वेंस डेटाबेस रिसर्च सर्चिंग हो सकती है और सीक्वेंस अलाइनमेंट। फंक्शनल एनालिसिस में मेटाबॉलिक पाथवे मॉडलिंग हो सकती है, जीन एक्सप्रेशन प्रोफिलिंग हो सकती है, प्रोटीन इंटरेक्शन प्रिडिक्शन हो सकता है और प्रोटीन सब सेलुलर लोकलाइजेशन प्रिडिक्शन हम इसमें देख सकते हैं। एंड देयर आर different software which are used to these prediction and these applications of its bioinformatics. Thank you for today's lecture.